Bismillahirrahmanirrahim. First of all, congratulations to AZSP team for organizing such a good event of 5th International Conference on Applied Zoology, abbreviated as ICAZ 2022. The topic of my presentation is estimation of selected metals in the blood of prostate cancer patients and reference. My name is Dr. Hafiz M. Abdul Qayyum. I am Assistant Professor in the Department of Chemistry, University of Education, Lahore, campus, Faisalabad. First of all, cancer. Cancer is not a single but group of diseases characterized by uncontrolled growth of cells. Cancer develops only in cells with damaged genes. It is a multifactorial and multi-mechanistic disease which can spread in any part of the body. Stepwise development of cancer involves different stages such as initiation, promotion, progression, and metastasis. Prostate cancer. Prostate cancer is one of the most common cancers in men. Screening for and management of early prostate cancer is one of the most challenging and controversial issues in all of the medicine. Risk factors. There are many risk factors involved to spread the prostate carcinoma. Number one is non-modifiable factors. Paramount among these is age. In unscreened population, prostate cancer has the steepest age incidence curve of all cancer and increases at approximately the sixth power of the age. Only 25% of the cancer are diagnosed before the age of 65 years. Genetic factors. The relative risk of developing prostate cancer is higher in men who have a first-degree relative with prostate cancer. This risk is higher in men under 65 years age. External exposure. Both ionizing radiation and ultraviolet radiation from the sun exposure have been linked to prostate cancer. Exposure to metals, including toxic metals such as aluminum, nickel, cadmium, mercury, and lead, have a major role in the development and pathogenesis of prostate carcinoma. In damaged DNA, three processes occur. Number one, gene mutation. Number two, inactivation of tumor suppressant gene. And number third is the activation of mutated gene. Due to above three processes, normal body regulation mechanism fail, and then coronal expansion and additional mutation takes place in the cells. Significance of selected metals. Essential metals are required for normal growth and development in human body, while their deficiency led to various ailments. Therefore, metals play both curative and preventive role in combating diseases. Trace metals are required at optimum concentration for proper functioning of the human biological system. Risk factors. There are many risk factors involved to spread the prostate carcinoma. Number one is non-modifiable factors. Paramount among these is age. In unscreened population, cadmium is non-essential for humans. It induces nephrotoxicity, immunotoxicity, and osteotoxicity. IARC classified cadmium as a group 1 human carcinogen, and cadmium develops cancer. External exposure. Both ionizing radiation and ultraviolet radiation from the sun exposure have been linked to prostate cancer. Exposure to metals, including toxic metals such as aluminum, nickel, cadmium, mercury, and lead, have. Copper occurs in more than 300 enzymes. Its toxicity may lead to prostate cancer, oral cancer, and ovarian cancer, but its deficiency may cause anemia, diabetes, and osteoporosis in children. Iron is mostly utilized in hemoglobin. However, its toxicity typically damages cells in the heart, Parkinson's disease, and develop liver, thyroid, lung, and stomach cancer as well. Mercury and its compounds are extremely toxic and must be handled with care. The most toxic form of mercury are organic compounds such as dimethylmercury and methylmercury. Mercury can cause both chronic and acute poisoning. Following are the major objectives of the study: to obtain a baseline data for selected essential and toxic elemental concentration in blood of prostate carcinoma patients with respect to healthy subjects, to investigate the mutual relationships among the essential toxic elements in terms of correlation coefficients, to find out the viable correlation among the essential toxic elemental levels in the blood of prostate carcinoma patients and controls, to find out the significant difference in concentrations of essential and toxic elements in various types and stages of prostate carcinoma in patients. Experimental methodology consists of following 4 steps: sample collection, sample preparation, meta-analysis, and the last one is the statistical analysis. Sample collection. The blood samples were collected on a volunteer basis from the newly diagnosed cancer patients admitted in Punjab Institute of Nuclear Medicine, Faisalabad. Sample from healthy subjects were also collected on a volunteer basis. Information regarding donors' diet, age, abort, smoking habits, health status, occupation, cancer type, and stage was also recorded on a proforma. Blood samples were collected through one veiny pincher method from an anticubital vein by using appropriate precautions to prevent exogenous contamination. Sample preparation or processing. The reaction was carried out on hot plate. 10 ml of nitric acid was added in a precisely weighed sample and then left for 5 minutes. After this, 10 ml of perchloric acid was also added and heating on hot plate at 80 degrees Celsius for 4 to 5 hours. Evolution of white dust fumes showed completion of digestion process. Final volume was adjusted with doubly distilled water. Statistical treatment of data involved minimum, maximum, mean, median, standard deviation, standard error, skewness, correlation, kurtosis, and quartile distribution is involved. Now the final results and discussion. Statistical distribution for the concentration in microgram per gram of selected metals in the blood of prostate cancer patient as shown in this table. On the average basis, average concentration of iron, magnesium, and cobalt were higher in the blood sample of prostate cancer patients. Similarly, statistical distribution of concentration of selected metals in the blood of healthy subjects as shown in this table. Average concentration of iron, magnesium, and cobalt were higher in the blood of healthy subjects. However, on comparative basis, average concentration of iron, magnesium, cobalt, chromium, and cadmium were higher in the blood of prostate cancer patients than the healthy subjects. Correlation coefficient matrix for selected metals in the blood sample of prostate cancer patient as shown in this table. This table shows that essential and toxic metals were correlated in the blood samples of prostate cancer patients. Likewise, correlation coefficient matrix for selected metals in the blood sample of healthy subjects as shown in this table. In this table, essential metals were also correlated with each other instead of toxic metals as shown in this table. Lastly, we discuss comparative evaluation based on demographic characteristics. Comparative average concentration of selected metals in the blood of prostate cancer patient and healthy subjects as shown in this figure. On the average basis, average concentration of cadmium, chromium, tin, mercury were highest in the blood of prostate cancer patient than healthy subjects. Comparative average concentration of selected metals in the blood of vegetarian and non-vegetarian patients and healthy subjects as shown in this figure. Average concentration of cadmium and mercury was highest in non-vegetarian patients as compared to non-vegetarian reference, whereas magnesium and cadmium was highest in vegetarian patients as compared to vegetarian controls. Comparative average concentration of selected metals in the blood of prostate cancer patients and healthy subjects based on smoking and non-smoking habits as shown in this figure. This figure shows that average concentration of cadmium, chromium and iron was highest in smoker patients than smoker controls. Similarly, mean levels of copper and iron was highest in non-smoker patients as compared to non-smoker controls. Levels of selected elements in the blood based on stages 1, 2, 3 and stage 4 of prostate cancer patient as shown in this figure. At stage 1, cadmium and cobalt were highest, whereas stage 2, nickel was highest in the blood of prostate cancer patients. Similarly, at stage 3, nickel was elevated, and stage 4, chromium, mercury and magnesium was highest in the blood of prostate cancer patients based on stages. Levels of selected elements in the blood based on types of prostate cancer patient as shown in this figure. In squamous cell carcinoma, magnesium, cobalt and chromium was highest as compared to other types of prostate cancer patients. In transitional cell carcinoma patients, iron and nickel were highest or elevated, whereas small cell carcinoma patients have cadmium, mercury and copper was highest in the blood of prostate cancer patients based on types of prostate carcinoma. Finally, conclusions. The average concentration of cobalt and cadmium were appreciably higher in the blood of prostate carcinoma patients than controls. Similarly, average levels of chromium and cadmium were significantly elevated in the blood of urban patients than urban controls. The selected metals like mercury and cadmium were maximum in the blood of non-vegetarian patients than non-vegetarian controls. In comparison of smoking and non-smoking habits, relatively higher levels of chromium and cadmium were elevated in the blood of patients with smoking habits, and magnesium and iron were maximum in the smoking controls. The correlation study revealed noticeably different mutual variations of metals in the blood of two donor groups, which evidences that the prostate carcinoma conditions were significantly affecting the metal imbalances in humans as well. Recommendations of the present study. The present study pertaining to select essential toxic metals linked with patients of prostate carcinoma. Various sessions are described below. Large-size clinical research should be conducted, especially considering long-term exposure of essential toxic metals. More randomized study should be done with large elements. Various biological specimens such as scalp hair, saliva, nails and milk should be used for diagnosis of this malignancy. Thank you.